It's my great pleasure and a great honor for me to introduce my good friend Berislav Zlokovic, incredible researchers, incredible physicians who actually is the head of the Department of Physiology and Neuroscience at the University of Southern California. Most of his work is dedicated to the brain-blood barrier, the cerebral blood vessels in AD, in Alzheimer's disease, and in stroke. He has identified the cellular and molecular mechanisms causing brain-blood barrier breakdown, for instance. He is one of the most cited researchers in the fields in the world for the last 20 years almost, since I knew he was incredible. And he got an incredible number of prizes: Potemkin Prize, Merit Award from NIA, the Javits Award from NINDS, USA Associate, a lot and a lot. And today we have an incredible chance to have him for all of us just in a presentation that will be outstanding as usual. So let me introduce Professor Zlokovic.
Bonjour, mesdames et messieurs. Je suis très heureux de faire partie de cette importante conférence. Je suis honoré d'avoir été invité comme un intervenant principal pour ces rencontres de la Fondation Alzheimer. Et je tiens à remercier mon ami, le professeur et directeur général Philippe Amway, pour son aimable invitation. Je suis impatient de discuter avec vous du rôle et contribution de défiance de la barrière hémato-encéphalique dans la neurodégénération, la défiance cognitive et la démence, un continuum qui inclut les individus atteints par la maladie d'Alzheimer. Je vais me focaliser sur les lignes entre APOE4 et la déficience cognitive et le rôle de la barrière hémato-encéphalique. Je discuterai aussi brièvement, si le temps me le permet, les opportunités thérapeutiques de ciblage de la barrière hémato-encéphalique pour ralentir, voire stopper le processus neurodégénératif et les changements cognitifs. J'inclurai des exemples basés sur la thérapie génique, ainsi que l'inhibition pharmacologique et biologique. À partir de maintenant, je vais continuer ma présentation en anglais. Je vous remercie de votre attention. Well, just as part of disclosure, I'm scientific founder of ZZ Biotech, a company with mission to develop new therapeutics for stroke and neurological disorders. So just to remember us a little bit about the physiology of the brain circulation. So perhaps, thank you, merci bien. So the brain receives about 20% of heart output, as you can see here, and spends about 20% of oxygen and glucose consumption. So it's only 2% of body mass, so it's very highly vascularized and has about 400 miles of vessels. So, but each this part of the vessels needs to be intact in order for certain brain regions to function. If it's get disrupted, then there is no good function of that part of the brain. So we do have a blood-brain barrier that develops in mammals, and this is a formation based on endothelial cells that are actually connected with the tight junctions, and also the green cells, pericytes over here, control the integrity of this blood-brain barrier. The blood-brain barrier prevents entry into the brain of blood-derived toxic products, such as, for example, fibrinogen, thrombin, and others, pathogens such as viruses, bacteria, fungi, as well as cells. So it has been found by different studies that in more than 50% of all dementias, we have small vessel disease of the brain. The people do have that, and associated with blood-brain barrier dysfunction. In Alzheimer's, Parkinson's, Huntington, multiple sclerosis, ALS. The key question that I have been studying for over, 35 years or more was, what is the role of blood-brain barrier dysfunction in neurodegenerative process, loss of cognition, dementia, and Alzheimer's disease? So a number of studies, animal studies, findings in rare human monogenic neurological disorders, and recent findings showing that blood-brain barrier breakdown is an early biomarker of human cognitive dysfunction actually shows convincingly that BBB leaks can lead to neuronal and synaptic dysfunction, injury, and neurodegeneration. So there are many pathways for this. So one of the pathways, red blood cells that go extravasated from blood vessels, and the brain doesn't like to have blood, it's a normal environment. So the mechanism is hemoglobin is released. Hemoglobin contains iron, le fer, and produces reactive oxygen species which are directly neurotoxic and also activate microglia. Fibrinogen is a molecule that activates normally microglia, but it also produces neuronal retraction and also disintegration and kills oligodendrocytes. Plasmin provides extracellular degrades extracellular matrix, so the neurons lose extracellular matrix and can die. And albumin causes edema as well. So we have then another pathway that can include autoantibodies, for example, here, that are against different neuronal receptors, and that can also play a role. Astrocytes get activated by albumin and produce TGF-beta pathway that is neurotoxic. And of course, loss of pericytes, as we've shown, leads to loss of neurotrophic support because of the growth factors that are produced by pericytes. So the blood-brain barrier, what I look at this, is like an ecosystem of the brain, or like what the ozone layer is for the Earth. Okay, so let me remind you, ozone layer is 20 to 30 miles above the Earth, and is only 3 millimeter thick, right? And so if there is a hole in ozone layer, then we get ultraviolet radiation from sun, no life on Earth. I look at a similar analogy with the blood-brain barrier. If we have holes in the blood-brain barrier, there is no good life for neurons. And there are about 10,000 transcripts and 5,000 proteins that have identified by recent multi-omic studies from different groups, including also our group. And just to illustrate the complexity of this blood-brain barrier system. So what blood-brain barrier normally does is maintain chemical composition necessary for normal neuronal functioning and provides most that brain needs for its metabolism, because a lot of things like glucose is not synthesized in brain. It has to come, from blood by a transporter called this GLUT1 transporter. So that's not why it's so strange that we have about 30 million copies of GLUT1 transporter around endothelial cells, same as much as we have histone copies for DNA that maintain nucleus. So the brain has to actually use its transporters for its metabolism, and I will illustrate with few examples, GLUT1, MSFD2A, for fatty unsaturated amino acids, and also one transporter, which is LRP1, lipoprotein receptor transporter. So I will just illustrate this with few examples, so the importance of this. So in one of earlier studies, we were actually asking ourselves what inherited loss of BBB endothelial GLUT1 transporter does to the brain. And my former, actually, PhD student, Winkler Eaton and Nishida, he is now assistant professor at Stanford, and this was in Tokai University, also assistant professor. They shown diminished glucose transport and blood-brain barrier breakdown 1 month in these mice that is haploinsufficient for GLUT1 transporter that is exclusively expressed at the blood-brain barrier. So this is followed by blood-brain barrier breakdown. But then with the time, which is about 6 months, neuron develop dysfunction, as we show by evoked cortical membrane potential responses to hind limb stimulation. So you can see this is a normal response of mouse that contains both genes for the glucose transporter. This is haploinsufficient mice that lack one gene, and you can see that actually by the colors over here, that the color is coming much, much later. So when you quantify, you see that these evoked cortical membrane potentials are completely distorted, which actually suggests neuronal dysfunction. And then when you do histology study, they found actually there is a loss in 16 months, about 15% loss in hippocampal neurons, just because of loss of this endothelial transporter, and somewhat less loss, but still significant at 6 months. And these mice develop microcephaly. So at 6 months of age, their brain is 4% less than what would be for a normal mouse that contains bulk copy. So it's very interesting because there is a human neurological disease that's called GLUT1 deficiency, and this also develops microcephaly developmental delay. So that tells you one example. You lose this transporter that is specific for sugar, and is present in endothelial cells, and the cool brain get compromised. So that's not the only example. There is another example of this transporter which transports essential omega-3 fatty acids. It's called MSFD2A transporter. So it's predominantly in brain, is mainly expressing or only expressing endothelial cells. So when you have a knockout mouse, this mouse develop leakages in brain, as you can see here, and also there is this loss of blood-brain barrier integrity, and missense mutation in humans leads to neurodegeneration and microcephaly and could be fatal also. So the baby cannot go over, maybe a couple of weeks. Now, another example is a lipoprotein receptor, an ApoE receptor, which is a major receptor for clearance of amyloid beta, alpha-synuclein, and tau from brain. So we were interested, when we started this study, we were interested to see how deletion of these receptors actually influence amyloid beta pathology and etc. But to our surprise, the first thing we found, there is blood-brain barrier breakdown at 2 months of age. So these mice who lost this receptor, lipoprotein receptor at the barrier, which is ApoE receptor, they have complete loss of blood-brain barrier integrity. And you can see here also shown by multiphoton or by tissue analysis for fibrinogen. So this is quantified here. You can see these huge increases in blood-brain barrier breakdown, and it's already apparent at early age. So since we routinely then look at the neuronal situation, we actually thought, okay, let's see if this has any effect on neurons. So at 2 months of age, neurons were absolutely fine, both in hippocampus and cortex, as you can see here. But at 4 months, there is a loss of neurons from cortex and hippocampus, and quite significant. If you quantify, it's about 15% of loss, or 15-20% of loss of neurons, and also related to actually behavioral abnormalities. So then we ask, okay, how about if we use gene therapy for blood-brain barrier dysfunction, and we deliver this gene back? Of course, this gene is huge, 600 kilodaltons, so you cannot do that, but you can do something else. There is a smaller mini gene, which is only 4th binding domain of there are 4 binding domains of this receptor. It's only 4th binding domain of the lipoprotein receptor, but has cytoplasmic domain that does signaling. So we get this specific AAV2BR, brain endothelial specific delivery of gene, because this AAV2BR affects, only endothelial cells, doesn't go anywhere else in the brain. So it's a specific, enrichment. And so what we were able to do, we were able to replete this mouse LRP1 with the mini gene by about 45 to 50% of vessels. So, okay, let's say, does this do anything? Could we really recover? And so we've done that. And then what we found is, that these vessels that did express these genes actually stop leaking so much. Not completely, not 100%, but 70-75% stop leaking. And then the careful analysis that Zhen Zhao did in my lab, it showed, so this is the vessel that expresses LRP1, and the part doesn't express. So the part doesn't express leak, the part that does express look okay. So we reduce leakages, and then we ask another question, whether reduction of leakages at 3 months of age, when we still don't see neuronal loss, can prevent loss of neurons between 3 and 4 months of age. And that was very interesting because, and I wouldn't say surprising because I saw this data now a long time, but I think we actually were able to also, by protecting blood-brain barrier, protect neuronal counts and also axonal density. And as you can see over here, and also behavior. So these are, we did 4 tests, but I cannot put it on slide because we're going to be too busy. You know, so we recover neurons, we recover we recover behavior. So that means if you act on the blood-brain barrier, in this case gene therapy, and you replenish what was lost, you know, then you can get actually also protection of neurons. We didn't know how long it will last, you know, but certainly we can see that this actually prevents and delays loss of neurons. And we have some other examples, but not time right now. So now, I think one of the best examples for those people who don't believe that blood-brain barrier dysfunction or loss causes some neurological disease are human rare monogenic neurologic diseases. There is about 20 of those. And so these are human diseases in which genetic defects is not found in neurons, but is found either in endothelial cells or in pericytes or in the basement membrane of the capillaries or vascular smooth muscle. So that's all kind of vascular cells. So I told you GLUT1 deficiency. So that gene that is in endothelial cells, the kids develop small brains, microcephaly, seizures, and have developmental delay. MSFD2A, intellectual speech disability, but missense mutation, many kids die because they don't have developed brain. They die on the week or 2 weeks, you know, depending on how serious is missense mutation when they were born. Then you have Allan-Hendron-Dudley syndrome, which is MST8, which transport thyroid hormones in brain. So that's another example. And then definitely tight junction proteins such as occludin, JAM3, CCM1, and basement membrane proteins that can lead to breakdown of the barrier and neurologic disorders. So now, if we know that, and then if we know also that there is blood-brain barrier breakdown in Alzheimer's disease that was shown by 20 or more independent studies, you know, like for example, fibrinogen that obviously comes from blood and goes with amyloid iron that is found in the middle of the plaques, you know, and you know, obviously comes from blood. Then the question becomes, is there any analogy between Alzheimer's disease and these human rare monogenic diseases, and when this problem starts? When it starts? Because pathology cannot tell us anything about when. So for us to address this, we developed in 2015 the modified dynamic contrast enhanced MRI technique that was used in multiple sclerosis, brain tumor, and stroke, but has to be modified to be adapted to study subtle blood-brain barrier changes. And we basically developed this method, and we studied first a population about 70 people, you know, different ages, with and without middle cognitive impairment. So the technique involves gadolinium injection, peripheral. It is about 15 minutes, and you follow on scan, 3T scan typically, or 7T if you want. And then you calculate these BBB-K trans maps for the entire brain. It's actually like a retinogram when you get, you know, for the eye with fluorescence. It's similar. It gives you, are there some leaky things or not? And so the first study that we published in 2015 with Axel Montagne, who is now in Edinburgh, has his independent group as a reader there, I think we followed people from 20 years old to about 90, and we found that there is leakage in hippocampus, mainly, hippocampal gyrus sometimes, but other brain regions are fine. These people are cognitively unimpaired. But there is tendency of the blood-brain barrier to start leaking. So when you then compare this, compared to young, so you can see the map here. When you then compare individuals with mild cognitive impairment, this leakage is considerably higher and coincides with this impairment. So obvious question was for us to ask, are A-beta and tau, which we know both vasculotoxic, are they really the reason for these leaks? So we've done a study that included about 200 and more patients, participants, that are actually either A-beta positive in CSF test and has a less A-beta in the CSF, or tau positive with the p-tau more than these accepted cutoff values. And then we actually did 10 different neuropsychological tests for memory, attention, executive function, language, global cognition, etc., usual stuff. And we found that really does not depend whether you're a beta 40 negative or positive. If you are at the stage of CDR0, of no domains impaired, then the blood-brain barrier is intact in the hippocampus. If you have one or plus domains impaired, the blood-brain barrier is getting to be leaky, regardless whether you're a beta 40 positive or negative. And similar happen in parahippocampal gyrus. And also, the same method for analysis showed that it doesn't really matter whether you're tau negative, p-tau negative, or p-tau positive. It just depends whether your domains are impaired or not. So we actually thought, well, look, here is the blood-brain barrier that show you a biomarker of cognitive dysfunction. And it's not really related so much whether you have a beta positivity or negativity. So it can happen both in individuals who are on Alzheimer's spectrum, individuals who are not. So now we go to ask about going to the ApoE and say whether the ApoE4 gene, which is the main susceptibility factor for Alzheimer, what is the effect of ApoE4 gene on this blood-brain barrier? Does it have effect on the blood-brain barrier? So you know that our human ancestors all have E4. So that was good at that time. But then when we start living longer and etc., that wasn't good. And then based on micronutrient diet and vitamin D, especially carotenoid diets, we got the first mutation. So that ApoE4 mutated in arginine 112 to cysteine 112, creating ApoE3. We know that ApoE3 carries now average risk for Alzheimer's. And then there was another mutation during evolution that this arginine 158 also was actually mutated to cysteine, and we get ApoE2 that actually has a protective effect if it is with E3 or E2. So, but E4 was still not extinct during the evolution, still remains the factor, and it's not rare. Actually, it's not rare at all. You have, for instance, depending on which population you look, but you have about 18% of people that have E3 and E4, and one E3 is not good enough, as you see, and then E4, E4 both are 5%. So our first study showed, actually that was recently published in Nature, showed that actually it's at a level where the people are cognitively unimpaired with a clinical dementia rating of 0, which does not mean that all neuropsychological tests are okay, still have open blood-brain barrier in E4, and when it progresses to clinical conversion into dementia in CDR.5, there is much more opening of the blood-brain barrier and leakage. Here shown for hippocampus, but we found the same situation in parahippocampal gyrus, and sometimes often in caudate also, but not other regions, not white matter, not cortex and stuff. So you can see this is a huge difference here, and also potentiated with E4 gene. Again, by CSF analysis, not dependent on A beta, whether positive or negative, or p-tau. So now, okay, CSF shows one thing. So in a subset of patients, we've done in parallel PET for amyloid, PET for tau, and we did our BBB test, and we did regional analysis. And then we found, this is like 74 amyloid patient, 96 tau, and we found that in hippocampus, no changes in amyloid accumulation or p-tau, no changes, but there is in this subset also an increase in blood-brain barrier breakdown, which then suggests, okay, that's not related to each other. And then we did other way analysis. We did orbital frontal cortex in E4 carrier is typically predilectional site for accumulation of amyloid. And then you can see this difference, in amyloid, but no difference in blood-brain barrier, suggesting these are two independent pathways. Now, what was also interesting, when we done hippocampus degeneration in quite a number of participants, over 230, we found that at the 0 stage in ApoE4, there is no change in hippocampal volume. And that is the phase when we already have opening of the blood-brain barrier, so which tells us that this blood-brain barrier opening happens before hippocampus get degenerated, and when the people move to clinical CDR 0.5. This was cross-analysis, so it wasn't longitudinal. We also show loss of hippocampal volume. So that actually is happening after loss of blood-brain barrier permeability. Now, we had a biomarkers, like 40 biomarkers, you know, for each cell type in the brain, and including pericytes. And why I'm showing pericytes, because that was one that was highlighted all the time. So this is soluble form of PDGFR beta receptors that get degraded by ADAM10 and ADAM17, and it's actually coming in the CSF when there is injury to pericytes. It could be hypoxic injury, it could be any other injury, and so you can measure it. And so we had about 350 participants, and then we asked, you know, at that time, what happens with these people? You know, we use a median split about 600, that's actually the level. What happens with those people who have high levels, blue, and who have low levels when they are studied by genotype? And so what we found, that APOE4 carriers, and this is now studied with a follow-up 2.5 to 4.5 years, that APOE4 carrier by mental status that was MoCA and MMC, they show progressive decline, 2.5 years, 4.5 years, compared to those who don't have increased levels. So these are those who have increased level, which tells you that this blood-brain barrier dysfunction because of pericyte loss can actually lead and predict to the loss of cognitive function. And same was shown on global composite with 10 cognitive tests. Now E3 didn't do that. You know, has a little modest effect. So now we went back to our original paper published in Nature 2012, in which we showed that there is E4, the mouse that is actually carrying E4 genetic risk does not have a bleeding, and has a bleeding, and those that does not have risk has intact BBB. And so we knew from atherosclerosis study that cyclophilin A is one that leads to the breakdown on the tilial barrier. So we showed the same by using a mouse that lacks cyclophilin A gene, crossed with APOE4 humanized mice. And if you do that, if you eliminate cyclophilin A, you got the mouse with no bleeding in brain, and with no, sorry, not bleeding, but with no blood-brain barrier breakdown. And also pharmacological blockade of cyclophilin A here with cyclosporin A or with Debio 25 results in the same thing, eliminates thrombin and fibrin from neuronal bodies. So that means that there is a way that if we act on cyclophilin A, we can protect the blood-brain barrier integrity. And of course, you know, there is effect of neuronal dysfunction that is similar. But here shows just neuronal dysfunction in E4s. So what was interesting, 10 years, 8 years after that, based on these findings, we also say, okay, well, we know that this pathway, cyclophilin A, in pericytes that leads to MMP9 production and degrades the blood-brain barrier that we discovered in 2012, well, we say, okay, but we can't do that study in humans, living humans, of course, but we can see what are the levels of cyclophilin A in CSF and MMP9. And sure enough, we found, to our pleasure, we found that there is increased level of cyclophilin A as the disease progresses to clinical progression, and also at the initial stage, both of cyclophilin A and actually MMP9. And since we already know that pericytes are injured, that actually makes us to think that there is similar activation of pro-inflammatory cyclophilin A and MMP9 pathways at the BBB, both in humans that carry APOE4 gene, similar as in humanized mice. Now, in a follow-up study that we did, we basically asked whether pharmacological inhibition of CPA-MMP9 pathway in pericytes in APOE4 mice crossed with Alzheimer's mice, what it does. And we used Debio 25 that was used in human hepatitis C, so it's been used in humans. We found no effect on amyloid, but we found that cyclophilin A is inhibited, so you don't see it in Debio animals. You see blockade on MMP9 pathway, and very interestingly, we were able to partially prevent further loss of neurons. Not completely, but at least to the level of E3s that are actually have amyloid there and loss of neurons, but we were able to do the same thing and show that we can recover, we can prevent some loss of neurons in E4 by acting on pharmacologically inhibited this pathway at the blood-brain barrier. And also some of the behavioral tests were showing the same situation. So now, you know, that's not the only thing. In most recent paper, when we talk if there is a blood-brain barrier, but let's go now, all right, well, we know cyclophilin A pathway is an important pathway, but is there something else? And then we recently done a paper and published this year, a few months ago, with brain endothelial transcriptome in E4 mice. So this is complete isolation of nuclei from brain of this mouse and RNA sequencing. And we found that both mice at 2 to 3 and 9 to 12 months have upregulated number of genes. If these genes that are upregulated in both is like about 140 genes are adhesion proteins, which are actually putting the vessels together, solute transporter, and link to cytoskeleton. Then in later stage, you have injurious DGs. And so that really shows the molecular signature of the failing blood-brain barrier. We've done phosphoproteome study also from brain capillaries in these mice. And the phosphoproteome study that was done by RNA-seq guided analysis from brain capillaries, or proteins from brain capillaries, showed that 50% of phosphatides, about 1,200, were dysregulated in capillary endothelial cells and in pericytes. Mainly phosphorylation of cytoskeletal proteins, DNA binding and RNA binding proteins was found in both cell types. But more in-depth analysis that used also phosphoproteome and proteome data, which means you know how much is the level of expression change, suggested that our expert in bioinformatics who did phosphoproteome and proteome analysis in Belgrade database came up with this diagram over here that shows that in the endothelial cells we have deeply disrupted cell junction and cytoskeleton proteins. The node that actually leads to that is TJP1 gene that is actually encoding zonal occludence protein 1, ZO1. And it's linked to all these different cytoskeleton genes and is also linked to all these different tie junction genes and adherence junction genes, as well as to clathrin-mediated transport. So I think this obviously is a target that we are focusing right now, whether with the gene therapy in some way we can act to prevent the situation. And in pericytes, we found that alternative splicing, RNA alternative splicing, is altered, suggestive of DNA damage and death, and obviously transcription regulation and nuclear export. So these were changes. What was interesting to us, we did interactome in these mice, postsynaptic density interactome. So we isolated postsynaptic densities and then did all these different proteins, hundreds and hundreds of proteins that interact with PSD95. And what we found, that at 2 months of age, when barrier is disrupted, all these nodes that are shown in gray are just completely intact. There are a couple of these, this is PSD95, there's a couple of these, only a couple that are moderately disrupted. If you look at 7 months, you have complete synaptic deficit, and you have all these pink nodes that actually show disrupted interaction at synapses of this PSD95 protein with 45 other proteins, including glutamate receptor, core scaffold machinery of PSD, and protein kinases. There was also loss of axonal density that starts appearing at 4 to 6 months, but all these changes are far after the blood-brain barrier breakdown, and as well as then behavioral changes that are occurring at 6 to 8 months of age. So there is delay then what, like maybe 4 to 5 months until the neurological complications develop in these mice. Now, I want to switch one other thing, because we've been studying in stroke 3K3-APC, which is activated protein C, which we mutated genetically to get the compound that loses anticoagulant activity, and is extremely vascular protective, anti-inflammatory, and neuronal protective. This was shown by dozens and dozens studies that are listed here. So I had a company based on that also, that I founded. And so we've done phase 1 stroke study, phase 2 completed successfully, and show that there is decrease in hemorrhage similar as in the mouse models. And then now we started phase 3 study in 1,400 stroke patients supported by NINDS and my company. And phase 2 study, LS, is just completed in Australia, so we are now waiting for the analysis and see what the results will show. So why I'm showing you this, this is the guy, Pat Leiden, who he was a chair of neurology at Cedars-Sinai. I was able to recruit him in my department now. He's leading this study. John Griffin is co-discoverer of this pathways with APC together with me, and this is just a CEO. All right, Kent. So I was always thinking when I went to Biogen to talk about, you know, all these antibody therapies and call the ARIAs and stuff. I said, guys, they call it ARIA is a fancy name for blood-brain barrier breakdown and vasogenic edema. Why we call it ARIA, you know? So, but I think wouldn't it be nice that you offer protection when you give the antibody, offer protection of vasculature, and maybe then the side effects would be less, you know, and probably. But, you know, it's complicated, so they never accepted that. But I think we studied this effect now in E4 mice, and we show that APC for 10 days, given at a small dose, is not completely, but is protecting blood-brain barrier breakdown in these mice. And you can see also in fibrinogen, so it's less fibrinogen, and also recovers the pericyte numbers. And interestingly enough, blocks cyclophilin A pathway. That is one of the pathways that play a role in breakdown. But these are unpublished data. But what was fantastic to us to see, you know, this is E4 mice compared to E3 mice. Everything was upregulated because there was abnormality. Everything is completely, by Volcano path, all genes, 165 genes have been deactivated by APC treatment. And you know, these are genes, the cell adhesion, solid transporter development. They were all downregulated, but normally they're upregulated, these mice. So, and more than that, there is 537 DGs, minus 167, so it's minus 350 that are also activated, which we're now looking about the protection. And then the phosphoprotein work has shown that you see in green nodes are those that are abnormal, but they are now, phosphorylation was normalized by 3-KT-APC. And there was a paper in PNS by another group who showed that it has a very prominent effect on phosphoproteome. So that means that the function of these networks that are involved with cytoskeleton, in angiogenesis, PI3-AKT, mTOR, VGF, and cell adhesion is actually coming back to normal. So these are genetic effect, transcriptome effect, and effect on the phosphorylation. And in the paper published by he is now promoted to associate professor, Abai, and she was a PhD. They show that APC actually, besides direct vascular protection, anti-beta protection, anti-inflammatory, inhibits BACE1 also, and inhibits BACE1 through NF-kappa B pathway. So that's also very interesting. So in addition, you have an effect of controlling amyloid pathology. All right. Now, I'm going to show you a few slides unpublished with our most recent intriguing interest about microbiome, because everybody's talking microbiome. We got one grant to study effect of microbiome, gut microbiome. And we just, you know, there are studies showing that ApoE influences gut microbiome. In mouse model, we confirmed that, and in ApoE4 carrier. The question I was asking in my group, is there a connection between ApoE gene effects on microbiome and increased BB permeability? And obviously, what is the effect on neurons? So these are published data, very intriguing. So we treated these mice 9 to 12 months of age, you know, E4, E3, and with antibiotics. There was a cocktail, amoxicillin and clavulinic acid, and they were given an autoclave tap water for 2 weeks. And then we've done MRI. We collected fecal pellet for metagenomic analysis, and we did an MRI at the end, and also CSF blood and all. So what was interesting that E3s, of course, they have intact barrier. You can see all blue, barrier is low. E4 have broken barrier, but antibiotic treatment closes the barrier. So how is that, you know? I mean, we still don't know. It's very puzzling, but it's normalization. And that was confirmed by histological studies, also by fibrinogen. And similar, based on our experience in other models, we also found that the neuritic density, axonal density, is improved by antibiotic treatment. So this is a short treatment. And then the metagenomic analysis have shown that both E3 and E4, this is single mouse, each single mouse, have similar number of bacterial families that are by antibiotics down, and the family by beta index showed that all families are, the same families are present in both. So what was interesting and what was different is that normally E3 mice before antibiotic treatment and E4 have a much more of this Bacteroidetes species and less Firmicutes species than E3 mice. So that was apparently quite significant. And of course, the antibiotic treatment brings them down. And the only bacteria that actually is in both present are Firmicutes. So now to get a little bit more in this, you can see that Bacteroidetes phylum, Bacteroidacea are up, you know, and then down over here, and all family order, etc. And then in Firmicutes, you have all these Clostridias, and Lachnospiraceae and Peptococcus are these families. But then when you treat them, there are other families of Firmicutes that come up. And, you know, and these are these two Streptococcus, and I'm sorry, I'm still learning, breaking my language on this, and Leuconostocaceae, okay? Sorry about that, you know. I have to practice more. But you know, what is interesting, there is a clear correlation between abundance of Bacteroidetes and breakdown of the barrier. Very clear, and Bacteroidacea. But then when you treat with antibiotics, this disappears. And the only thing that are 100% present is Firmicutes and no effect of barrier. So what we know, that Firmicutes do not damage the barrier. And now we are trying to do experiment to eliminate Firmicutes and see if barrier is going to be open to see if Firmicutes are protective. So that's actually would be really, really nice if we can show that. And so what else I have? And of course, then this pathway by antibiotics was shut down. We look at first encyclophilin A and FKB MMP9 pathway, and this is just a few 5 to 6 preliminary studies unpublished, but it's exciting that CPA and FKB and MMP9 all get down regulated. Now, one can ask, is antibiotic directly affect blood-brain barrier? Is it, you know, we don't know. So what we've done, we use iPSC-derived parasite-like cells that we publish in Nature 2020 that secrete a lot of cyclophilin A and produce MMP9, and we treated this with the same antibiotics for 5 days with a concentration that in human subject, based on pharmacokinetic data, is actually the maximal concentration, and no effect on cyclophilin A levels. So we know there is something else. There is work in progress. I didn't want to show because it's too preliminary, but I tell you, we are thinking about some cytokines and chemokines, and we are finding one of these that is actually altered in E4 mice, and that can represent a target. So I don't want to say anything because I don't know, maybe I'm not going to be right. We have repeating these experiments right now. So what I showed you to summarize key points is that the BBB, I'd like to use this ecosystem like ozone layer comparison, is critical for normal brain metabolism and functioning. BBB dysfunction in transporter genetic defects leads to CNS dysfunction. And we have human monogenic disorders and models of this disorder that show no question that you have neuronal problems after blood-brain barrier problems. BBB breakdown is early biomarker of human cognitive dysfunction. We show that in few papers. Then APOE4 carrier are distinguished from APOE3 homozygotes by preclinical BBB breakdown. So this is the preclinical stage in unimpaired, cognitively unimpaired. In the hippocampus and medial temporal lobe, which becomes more severe with cognitive impairment, with clinical progression, but is not related to a beta tau pathology. So it could be found both in those who have and those who don't have. And BBB dysfunction can predict cognitive decline in APOE4 carrier and precede synaptic dysfunction in E4F mice. Now, therapeutic approaches directed at BBB can slow down and possibly prevent development of neurodegeneration. We certainly know it's slowing down. But how long, we don't know, but it's definitely there. So this is brain endothelial AAV gene therapy. I show you example with LRP1. We also have examples with PCOM and GLUT1 transporter. Pharmacological inhibition of cyclophilin A and MMP9 pathway in parasites improves vascular and neurodegenerative disorders in humanized APOE4 FAD mice in the presence of advanced A-beta pathology. So that's really important clinical application because, you know, even if you have advanced pathology, you act on blood-brain barrier, you can still have some improvements. And biologics such as our APC that is in phase 3 pivotal trial for stroke, in phase 2 proof of concept studies completed, has potential to fix BB damage, especially in situation of antibody delivery, offer neuroprotection and reduce inflammation. Now, ApoE4 effects on gut microbiome, we are still studying, does this play a role in ApoE4 mediated BB dysfunction? We think yes, but it's still in works. So this is my group, my support from Institute of Aging, NINDS, Alzheimer's Association, Cure Alzheimer's Fund, Leduc, Open Philanthropy. How much I supported, I get ashamed I didn't do better. You know, I should have done better a little bit and find something more things. But anyway, it's been really great to be supported. And this is our microbiome team, you know, which is Mingzi Zhang, postdoctoral fellow, Cassandra Kissler, she is actually a research assistant professor, Mikko Huskonen, he came back to Finland, Ararat Chakoyan, he's from France, you know, he's assistant professor now, he's associate professor at BI, Mariangela was part of the project, went to industry for a good position, Yaming Wang is our modelist, stroke modelist, and Marcello Koba is bioinformatics protein guy, and Karina. So thank you very much for your attention. Merci bien. Why is the BBB breaking down? Well, I think, do you want to, I tell you my theory? Yeah, that I have 20 years ago when I started doing genomic studies. I think that basically the genomic or transcriptional, you know, composition or gene composition of the BBB has much less longevity than neuronal. And that we were actually, when we first started doing that in 2020, we were doing Affymetrics and stuff, studies in human pathological samples. We found, you know, that there is a really change in gene composition during normal aging, and there is changes in this composition. Why this change is happening, it's not related to telomerase. I was spoken with this lady who got Nobel Prize for telomerase. We don't think it's telomerase. What we think is the genetic code is not forever, because if it is forever, we would live forever. But still, we're not at that stage. So I think that this proportion of the code that is given to vascular cells and neurons is actually leading to this. Right now, you have other theories that you see Mediterranean diet, you know, Western diet is not really good. And there is a lot of other things that we don't good for the brain. And there is evolutionary theory that is very difficult for the vascular system to maintain. If you accept that our life started in the ocean, and then we get back to the oxygen to conquer the Earth, and then we're going to conquer the Mars or Moon or anything, it's very challenging for humans. Humans not ready. I spoke with astronauts who spent 99 days. They cannot get back to normal after a year. The kidney function, we are not perfect organism for that. And we like to be. And so that is related to diseases, especially the vascular system everywhere. Why is the heart, may I ask you? Vascular system. Why is the veins in the legs? Vascular system. So I believe that's probably an answer that I would give.
Thank you very much for the fascinating talk. Sorry.
Yeah, we did lipidomic studies, complete lipidomic studies, and we found some interesting changes, but I don't think we found conclusive changes there. So because of that, we got back to cytokines, and we have a cytokine candidate, but I'm hesitant to tell you which one, because I don't want to be wrong. But we have it. We have it. Mm-hmm. 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 Yes, yes. Mm-hmm. Mm. Well, I think this is an excellent question. You know, what you're asking. We published Donation in Science with Winkler and Ethan Winkler, my former student who is now at Stanford. We just published this year in April. But taking the big vessels also, so which has different composition, because we were interested in a totally different question. You know, we were interested in arteriovenous malformations in that paper. I mean, he was interested. I was just secondary helping. And you know, there are definitely genetic changes that do affect vasculature. Because of that, vasculature gets compromised. And in AV malformation, that's obviously the case. Now, regarding your question, you know, I did discuss with Christer a lot that, you know, we see each other. He was just visiting with me privately in October, and we have all this discussion. We exchange all ideas and stuff. And, you know, it's not really clear, honestly, what is the, you know, the first belief was, based on the mouse studies from Christer data, that, you know, there is endothelial cells everywhere are the same. Well, I disagree completely with that, you know, and I think there is some differences. I discussed with the late Paul Gringard, because Paul, in his 91, was interested in the blood-brain barrier. We met in Rockefeller, and he was doing certain studies. He first also was not thinking about regionality, but then the people start thinking more. So I do believe that there is the regional differences. So now, you can get from what I'm trying to do right now, I'm getting from epilepsy surgeries, the part of the hippocampus and cortex, and I'm trying to do proteome analysis because, you know, RNA-seq analysis is a little bit more difficult, could be affected by the length of the procedure and everything else. And we are doing that at this point. So, you know, the surgeons are giving us the part of the cortex that it should be normal and the part of the hippocampus. I personally do believe that there will be differences in proteomic composition, etc. And, you know, first of all, hippocampus phylogenetically is happening, was developed earlier. So it may not be perfect, you know, and the neocortex was developed later, you know, when we, in higher mammals. So I think there will be regional differences. I mean, the obvious differences are between circumventricular organs and other organs, but it's much more complex picture. If you talk about neurons, okay, everybody will agree, yeah, sure. Neuron hippocampus, of course, difference from neuron cerebellum, of course, difference from neuron's cortex. But you think the vessels are the same? That's a joke to me, you know. I'm sorry. It's my bias. I do believe the regional difference, but I don't have a proof, you know. But in animal models and humans are the same thing. We see hippocampus, parahippocampus gyrus, for some reason to collapse. Unfortunately, these regions are important for our cognition, learning, and memory. If it happens somewhere in the cerebellum, we'd probably be walking like this. Or if it happens in the motor cortex, we also have this theory in ALS. It could be very similar. So, but why? You know, I mean, I hope that we'll be able to show. Because in mouse, you can't do that. Because the amount of tissue that you take is so little. You have to kill 100 mice. And put, so my IRB will never do that because say you don't have a reason to do that, you know. Oui.
Thanks a lot for the talk. I still have some difficulty to believe that ATP and médecins probably not involved in the breakdown of the blood-brain barrier. When we see the damage which there might be some, you know, some of the considerations that might be more soluble that you don't see on the page, but it could be more.
Yeah.
What is your
Let me clarify this, you know, because I haven't discussed this, because I was particularly interested in initial stages of preclinical cognitive impairment and clinical progression, which is very initial stage. And we've done beta oligomers, we've done tau oligomers for that paper, and there was no correlation. But I'm not saying that at the later stage, when you have CAA, you have an additional blood-brain barrier breakdown. So there are two stages. The first stage that happens very early and is not related to either amyloid or a beta. You have later stage when you have a lot of amyloid deposition, and of course is related to vessels, you know, hemorrhage and also the small capillary hemorrhage. We see those. We see microhemorrhage with 70 about in 75% of, you know, people with APOE4 that are CDR actually moving to CDR 0.5. So please don't misunderstood what I'm saying. A beta and tau are vascular toxic, and at certain stages when they accumulate, they have very strong vascular effects. But what was interesting, what was fascinating to me, that in these initial stages, you know, we don't see dependence on either of these factors or association. Did it clarify?
yes, but
Okay. Merci bien. Oui.
Hi, I'm expert of your presentation. And regarding this aging effect, do you know if there's a correlation of the BBB detection and the cognitive reserve
Mm-hmm.
normal aging?
Yeah. Excellent question. Excellent question. You know, we're studying this right now. Right now, we have, you see, the first study of 5 years, we were not able to do many longitudinal studies. Now we got possibility to study them for another 5 years. So we are now recruiting the same patients in 3 sites at USC, Washington University, ADRC, and Banner and Mayo Clinic. They are recruited and with the same patients for follow-up points. And we study in all these patients BBB status, biomarkers, amyloid, and tau, and how they interact, and what it does cause. Now, in preclinical, what you're talking about, the preclinical cognitive impairment, we are doing these tests, you know, different preclinical tests, and we already know that cognitively unimpaired may have some problems in some memory tests and other tests that are refined, but we don't see it on CDRs, on cognition, on a clinical dementia scale. You know, it's not so sensitive like these different tests. So I have a wonderful neuropsychologist who work with us, and we are now trying to relay these changes. And then you can follow really the role in preclinical decline, which is preclinical impairment, and then you can follow them in a clinical. So the cohort of our patients is about, right now, participants is about, I don't want to throw a number, but I think it's close to 600. And 75% of theirs enroll as a cognitively unimpaired. But now it's already 5-6 years they follow, so they start now showing certain changes, and so they are moving. And those that were MCI, they were 20-25%, they're now moving also. So we have to be very careful now in analysis, we're collecting data. Sure.
Thank you for your fascinating presentation.
Merci bien.
And I have just a curiosity question. What could trigger the cyclophilin A pathway in the brain cells in a normal body?
That's an excellent question that we've been asking too. You know, I have an answer for you, but that goes to these cytokines that somebody asked me who I don't want to disclose, because we know that these cytokines does that. So there is a peripheral cytokine. Can I say that? It's peripheral soluble cytokine. And if you neutralize this cytokine, you actually lose much of cyclophilin A also. So there is a cascade, pro-inflammatory cascade that comes from blood to blood-brain barrier that actually activates this. But very little is known about, transcriptional regulation of cyclophilin A. I mean, in basic studies, basic science, very little is known. You know, so, but this is just one of our observations that I can say. So there is a reason why, and, you know, obviously, you know, we think that we know, but I think studies in male mice are in progress, studies in female mice are in progress, they may be different, so I don't want to say anything, you know.
Thank you very much for this.
Thank you.